I've really enjoyed the session so far, and I think everyone's probably enjoying not having to stand at the door or sit on the floor. It's kind of refreshing to have a sort of a normal occupancy for one of these sessions. So I'm going to be talking about type 1 diabetes and gene therapy approaches to type 1 diabetes, which are really, believe, it's becoming actionable to think about doing this in the clinic and not too distant point in the future. So type 1 diabetes has a genetic component, but it's not a monogenic inherited disease. It's an autoimmune disorder. It's associated with certain HLA haplotypes. And all of you here know, I'm not going to give a long introduction on type 1 diabetes. It's an autoimmune mediated destruction of the insulin producing beta cells in the pancreas, and therefore the way to improve the disease is to restore the existence of insulin secreting beta cells and modulate the autoimmunity. So just to start out with, because I know that's going to be a question if I don't address it, what we're proposing currently addresses only the generation of new beta cells, not the autoimmunity, and I think both will be needed for a definitive cure of type 1. So it's a pretty common disease, and for reasons people don't really understand, it's becoming more common in the Western world. Type 1 diabetes, the autoimmune type, is the major type of diabetes in young people, and it's going up 2 to 5% per year. There's about 1 in 300 people in the US have this, so you're looking at a total patient population of 1 million people who have insulin-dependent type 1 diabetes in the United States alone. The current standard of therapy is, of course, insulin injection, either with pumps or with syringes, measuring glucose all the time and so forth. And while that works, people really have been thinking about more permanent solutions. There's also a subset of patients who have insulin-induced hypoglycemia that they can't feel, and it's a bad problem. So in type 1 diabetes, you don't have the problem of high insulin, but some patients have recurrent episodes of hypoglycemia, which are very dangerous. And for that reason, the group of Shapiro in Edmonton, Canada, a few years ago now, decided to explore the concept of islet transplantation in an immune suppressed setting. So it's like an organ transplantation for diabetes, and it's still used today in patients who have this dangerous hypoglycemia type 1 diabetes, and patients can become insulin independent. Of course, they're also immune suppressed because it's an allograft, and so the autoimmunity is also suppressed in that setting. So encouraged by this success in this patient cohort, a lot of people have been thinking about how could you cure type 1 diabetes or replace the beta cells. And sort of the predominant paradigm, and there's several companies in this space, is to make insulin-producing beta cells de novo from pluripotent precursors or from somatic cells, and then transplant them back into the patient, again with immunosuppression or encapsulation, so very similar to what was done in the Edmonton protocol. And then what's coming on the scene, and what I want to convince you probably could work or should be further explored at least, is the idea of making new beta cells by gene therapy. And various papers, I won't have time to go all into them. It started with Sarah Ferber in Israel, showing that if she put pancreatic transcription factors into the liver, that she could get correction of hyperglycemia. And then various other groups have been exploring other targets for transcription factor mediated reprogramming to create de novo in situ insulin producing beta-like cells. So very briefly, I will recapitulate some efforts that we made in my lab on this. We targeted the gallbladder. We decided we would do the gallbladder because, as you can see on the far right, there's evidence from developmental biology. For example, you knock out HES1, which is in the Notch pathway in mice, and you can start getting insulin-producing cells in the gallbladder. And it's a dispensable organ, so we thought if we could pull out the gallbladder from patients, reprogram them to beta-like cells in vitro and transplant them back, that would have some advantages over embryonic stem cells. It would be an autologous transplant. We wouldn't have the tumor propensity and so forth. So we did this. We were able to expand cultured human gallbladder cells extensively. They grow very well, and then transduce them in vitro with a gene therapy vector. This was an adenoviral vector ex vivo, expressing 4 different pancreatic transcription factors, PDX1, MAF-A, neurogenin 3, and PAX6. PDX1 and MAF-A I want to call your attention to because those have turned out to be key transcription factors that work in multiple settings, and neurogenin 3 is also important. PAX6 is sort of fallen by the wayside. So when we did this and combined this in sort of with an embryonic stem cell-like differentiation paradigm, we were able to get insulin-producing cells from the human gallbladder. And if you look on the right thing at a heat map of human gallbladder cells on the left, real human beta cells on the right, and then reprogrammed gallbladder cells in the middle, you can see that extensive reprogramming towards the insulin-producing beta cell fate was achieved, and we actually got glucose responsive insulin secretion and things that you would be looking for in a real beta cell. However, despite the success, we were not really to translate that into an effective correction of diabetes in mice. The cells were not very stable after reprogramming, and transplantation of these cells was very inefficient, and we lost the majority of the cells, and we tried just about everything in the book that you can think of. So we have given up on ex vivo reprogramming of endodermal relatives of beta cells and have switched to in vivo reprogramming. So the idea here is, first of all, can it be done? Which cell type in vivo is the best target? What vector system to use to deliver the transcription factors? What exactly do you need in terms of reprogramming factors? And then is it safe? Basically, I've made the joke many times that if you want to have insulin-producing cells that correct hyperglycemia, just express the insulin gene in the liver or in the muscle, and you will get insulin, and blood sugar will drop dramatically, but it's not regulated at all, and you will create hypoglycemia and death from hypoglycemia. So it's not enough to make insulin. It's key to make glucose-regulated insulin-secreting cells, and the more important feature even is that they shut off, that they don't keep making insulin when the blood sugars drop below a certain threshold. So that needs to be achieved. So through a series of attempts of doing this, we have homed in on the ductal system of the pancreas as a way to achieve gene therapy in vivo. The hepatobiliary system consists of ducts that come from the liver, the bile ducts, and then a gallbladder attached to that, and the pancreatic ducts come from the pancreas, and they come together and empty their contents into the duodenum. When you look at what might be the target cell, we took advantage of RNA-seq, and we looked at this is a 2 principal component analysis of RNA-seq, see which of the cells that we might program are the most similar to pancreatic beta cells. And it turns out it's interesting, even though liver ducts and pancreatic ducts are morphologically extremely similar and they have a similar embryonic origin, the pancreatic ducts are really quite more similar to the pancreatic beta cells. Oops. So I don't know whether I'm going to be able to show the movie, but suffice it to say, we decided to start injecting gene therapy vectors into the ductal system, directing our reprogramming factors to the liver or the pancreas. And I want to make a pitch for intraductal delivery. Many times people use the vascular system to target a specific organ, and of course the problem with the vascular system is that it has an entrance and an exit, and blood goes through organs pretty quickly. In contrast, the ductal system of organs such as the liver or the salivary gland or the pancreas doesn't have an exit. So whatever you put in there is going to stay in there for an extended period of time, and the effective local dose of your gene therapy vector is going to be very high. So we injected in the pancreas and the liver ducts and looked for insulin-producing beta-like cells, and you can see that indeed we get reprogramming of cholangiocytes. You can see the red cholangiocytes there. EpCAM is a marker for ducts in general. And in the pancreas, we can also get them. Interestingly, we could not achieve reprogramming in the gallbladder with the same vector. So, but liver ducts and pancreatic ducts work quite well. But really, the pancreatic ducts responded to the reprogramming much better than the liver ducts. So I already mentioned that you don't want just insulin production. You want to really reprogram the cells more towards a more complete beta fate. And for that, you can look at other genes that are not direct transcriptional targets of your reprogramming factors. And one such gene is NKX6.1, important for the beta cell state, but not a direct target of any of the factors we used. And when you reprogram pancreatic ducts, you do induce NKX6.1, and it's properly localized in the nucleus in the majority of the cells. So this highlighted here a number of genes that were more beta-like when you reprogram pancreatic ducts than liver ducts. NeuroD1 is another one, and these two lower genes are associated with secretion of insulin granules. So that was good. Another thing was that in some of the reprogramming paradigms, including the gallbladder paradigm, when you use these three transcription factors, PDX1, MAF-A, and neurogenin-3, you actually generate multi-hormonal cells. You don't just get insulin, you get somatostatin and you get glucagon. However, reprogrammed pancreatic ducts make only insulin. So if we stain reprogrammed animals' pancreata with, for example, glucagon and insulin, glucagon in green and insulin in red, you can see the reprogrammed insulin-positive ducts in red. And there's an islet in there as a control. And you can see that there's no glucagon co-expression in these reprogrammed cells, and similarly there is no somatostatin co-expression. Okay, so it looked pretty good at this level, and so obviously the functional test is to see whether we can cure diabetes with this approach. We use several diabetes models. I'm just going to talk about this one, where you use a diphtheria toxin that's a receptor that's under the transcriptional control of the insulin promoter to drive diphtheria toxin receptor, and you can wipe out the beta cells by giving this diphtheria toxin. And here's what happens. The mice become severely diabetic. The reason they all peak at 600 is because the glucometer's top number is 600, and it's probably higher than that. But if you treat the animals with intraductal delivery of the reprogramming factors, in about 35% of the animals we have a complete and sustained rescue of the hyperglycemia. Depending, it's quite difficult to inject the pancreatic duct of mice efficiently. And so if the injection doesn't go so well, we get either partial reprogramming or no reprogramming. But here's what the data look overall for two models: complete correction, 35% with the RIP-DTR diabetes model, and streptozotocin-induced diabetes is the other model used there. It was about 40%. And again, the failures are related to difficulty of getting the full volume of the vector into the pancreatic duct. So I already mentioned, so the important thing is we didn't see any hypoglycemia, and another way to ask whether you have proper beta cells is to do a glucose response test. So the diabetic mice obviously have sustained hyperglycemia, and wild-type animals, after you give them glucose, will have a peak in glucose and then it'll come down, and our reprogrammed ductal-corrected mice had the same glucose response curve as wild-type mice. This is really a pretty good indicator of a functional glucose-responsive beta cell that also doesn't induce hypoglycemia. So functionally, they look pretty good. So are they really true beta cells? To do this, we did single-cell RNA-seq. And in these animals, we're not diabetic because we wanted proper beta cells from the exact same animal. So we just injected the reprogramming factor into MIP-GFP mice, in which insulin-producing beta cells are GFP positive. The transgene is driven by the insulin promoter. And we found 2 clusters of cells that express GFP, express insulin, cluster 1 and cluster 2. It turns out that cluster 2 is the reprogrammed duct cells, and cluster 1 are real beta cells. They both make GFP, they both make insulin. But when you look at the insulin intensity, they actually make less insulin RNA per cell than real beta cells. They do make NKX 6.1, and they've silenced ductal markers such as SOX9. The bottom line is, when we look at the entire transcriptome, they are highly beta-like, but not completely beta-like. And one of the differences is, for example, the level of insulin expression, and there are other genes that are not fully corrected by this approach. So conclusion in this part of the talk is that using an in vivo gene therapy approach, we can target and reprogram pancreatic ducts to monohormonal beta-like cells. They are able to correct multiple diabetes models in mice. And just, again, it's easier to do a ductal injection in humans than it is in mice. ERCP, or retrograde injection of the pancreas and the liver, are done by the gastroenterologists every day, and it's an established delivery method or diagnostic method in humans. So I think this could be done. You could put gene therapy vectors up the duct in humans quite efficiently. So the alpha cells, the ducts gave us pretty good result, but the prime target nowadays through work that was initiated by Pedro Herrera in Switzerland is really the alpha cell. The alpha cell is, of course, the cousin of the beta cell. It sits in the islet just like the beta cells, and it makes glucagon instead of insulin. But in many aspects, it's highly similar to a beta cell already. And what Pedro showed was that if you ablate the beta cells in mice, the alpha cells will spontaneously convert into functional beta cells in that setting. He had a Nature paper on that about 7 or 8 years ago. Others have taken advantage of that to target the alpha cell specifically for reprogramming, and a really nice paper came out of the Pittsburgh group last year by Giddis et al., in which they put an AAV vector. So they moved away from adenovirus, which we used, to AAV, intraductal delivery of an AAV vector that expresses PDX1 and MAFA, so only 2 transcription factors. We used 3 transcription factors. And they used the autoimmune diabetes model, which is the NOD mouse. And they got sustained correction of diabetes in that model. You can read that up. Then they also put this, the reprogramming factors on human cultured islets, and they could also see a conversion of human alpha cells in culture into beta-like cells. We, working with Klaus Kessner at University of Pennsylvania, had similar results. We didn't use in that experiment gene therapy. We basically used epigenetic modifier drugs and could show that alpha cells are poised at transcriptional start sites to be converted to beta cells, and then if you do the appropriate chromatin modifications, you can push the cells towards becoming beta cells. And so here's a summary of what the Giddis group did. So you have a mouse that has very few beta cells, and then intraductal infusion of AAV, you get reprogramming and euglycemia. Just this year, collaboration with Pedro Herrera, this was done with human alpha cells, and the difference to the previous experiments with human alpha cells is this time we actually went and corrected diabetes. So reprogramming factors were put on FACS-purified human alpha cells. these cells were reaggregated into pseudo-islets and then transplanted into diabetic mice. So here's an experimental outline of this paper that came out in Nature just a couple months ago, where basically you do ex vivo reprogramming of human alpha cells, then put them into pseudo-islets and transplant them into diabetic mice. Here's sort of the experimental outline of what was done there. And then they used the RIP-DTR mouse. And when you transplant intact human islets, that's the blue lines here, you correct the hyperglycemia. That's sort of the equivalent of the Edmonton transplant protocol. But the alpha cells that were reprogrammed correct the diabetes just as efficiently. So this basically is a proof of principle that human alpha cells, if reprogrammed by these factors, will actually function as proper beta cells. So this is all great data. So how can we move this forward towards the clinic? There's several things that need to happen. One is we need a better vector. The adenovirus, which works great in preclinical models in the mice, is not suitable for the clinic, so it needs to be AAV. And we need precise delivery to the target cell, either alpha cells or ducts. So, for example, the beautiful paper from Pittsburgh, they used an AAV that had an ubiquitous promoter, so it will hit any cell it transduces and express these factors. This is not desirable. You don't want off-target effects from converting other cell types into towards the pancreatic beta lineage. So the other issue is, which AAV serotype to use, and we have generated data in collaboration with Hiro Nakai on which are the best serotypes for human pancreatic endocrine cells. Hiro has developed a really cool method which is based on RNA, DNA barcoding of different capsids to ask the question which capsid gives you the best transduction. So the way these experiments are done, you put in a library of barcoded capsids, usually around 100 different serotypes, and then you fact sort out the cells of interest and basically look at the DNA barcodes to normalize for vector copy number, and then RNA barcodes to look for transduction and expression. And so we do this in human islets. We were looking for the best serotype for reprogramming. You can see that AAV8, which is the one that was used in the mouse studies, is about 2 logs less efficient at transducing human endocrine cells than these other vectors. AAV2 is pretty good. AAV3 and AAV LK03 were the best. And AAV DJ, which is on here somewhere, is also quite good. So with using that as a starting point, we had a collaboration with Mark Kay and Katja Patroon from his lab to use directed evolution for human islet targeting. And basically, these shuffled AAV capsid shuffled libraries were passaged serially over cultured human islets to look for serotypes that were really good at transducing beta cells in particular. Mark's lab did a 10-parent library and an 18-parent library. And out of this, 3 serotypes came that are novel serotypes that he talked about at this meeting that are really excellent at transducing islets. And the amazing thing is, I'll show you the data in a second, that these new capsids have the ability to penetrate into the core of avascular clumps of cells. So cultured human islets don't have a blood supply. And there are about 1,000 to 1,500 cells in each islet. So if you have high efficiency transduction, it means that your AAV went through multiple cells to the core of the islet and transduced everything along the way. Okay, so here's what it looks like. DJ and LK03 came out ahead based on that RNA barcode screen. You can see they're pretty good at transducing human islets in culture. 10A1 is KP1 and 18A1 is KP3, also highly efficient at transducing islets. When you look at these clumps, you can't see what percent the cells is transduced. So we basically took these islets and FACS sorted and looked for GFP expression. And here is 4 serotypes. You look at all islet cells, AAV-DJ, 50% AAV-LK03, 60%, the 2 neuroserotypes only over 90%. Beta cells, over 90% for marked serotypes, about 50% at an MOI of 100,000 for the other serotypes, and alpha cells 99%. There's never been a vector in diabetes gene therapy that has had this kind of performance before. That was at maximum MOI. If you dial back the MOI, you can see the difference between the new serotypes and the ones that the K-Lab generated more clearly, and there is no question that both KP1 and KP3 are about a log better at transducing human islets than the existing serotypes. Okay, so we think we now have a pretty decent human endocrine AAV capsid. Then the next thing is to make sure that we can reprogram only the target cell. Even with, AAV capsids, even if you evolve them for a specific cell type, they usually will transduce other cell types along the way, and certainly these new capsids that the K-Lab has are really good at transducing hepatocytes and neurons and all sorts of other cell types as well. So if we use them, even with intraductal delivery, we will be transducing acinar cells, ducts, and everything, and really our target in this setup would be the alpha cell. So my lab has been using the Nakai method, which is RNA-DNA barcoding, to explore and generate cell type specific transcriptional control elements. So the idea is that the AAV genomes get in everywhere, but the expression is limited to the cell type of interest. And the way we do this is we basically shuffle libraries of potential promoters upstream of an RNA barcode and then transduce human islets. So here's how it's done. We transduce human islets with a library. Our promoter library has about 1,000 different promoters in it. We FACS sort the alpha and the beta cells, and then we look for promoters that give us the property you want, which in the experiments we've done to date has been beta cell specific expression. We are only now beginning to work on the alpha cell expression. To do this, we deconstructed the human insulin promoter. We took all the major transcription factor binding sites from the human insulin promoter, made oligos for PDX1 and all these, shuffled them, and then put them in front of an RNA DNA barcode. It's important, of course, there are cell type specific transcription factor binding sites, such as for PDX1 and islet1 and so forth, but it's really important when you build a good promoter to also have ubiquitous binding sites, such as CREB binding site and so forth. So it can't just be cell type specific binding sites that you shuffle. So we did this and have generated a series of very compact promoters that consist of shuffled insulin promoter transcription factor binding sites that drive expression to levels that are similar to the CMV promoter, so very high levels of expression, even though these promoters are tiny, only 200 to 300 bases. And here you can see a few examples. Multiple PDX binding sites is pretty typical in these. They all have a NeuroD binding site in them and so forth. And it's really cool to be able to screen thousands of promoters in a single experiment like this. So even though they have enriched expression in beta cells, most of them are not completely clean, so they also express in other endocrine cell types. And to make them more cell type tight, we borrowed a trick from the Naldini group. We essentially took advantage of microRNAs that are differentially expressed in human alpha cells and beta cells, and we included microRNA binding elements that are highly expressed in alpha cells, for microRNAs that are highly expressed in alpha cells, and the idea is to basically silence leaky expression in alpha cells. And as you can see here, we're looking at RFP expression in C-peptide expressing cells on the top, and in glucagon expressing cells at the right, and you can see the alpha cells and the beta cells by virtue of their C-peptide and glucagon expression. And if you look at the far left, this is an insulin type promoter to demonstrate this. You have more expression in beta cells than in alpha cells, but there's plenty of RFP in alpha cells. And then you add in these microRNA binding sites, and on the far right you can see that pretty much the expression is now limited to only beta cells. So we have beta cell specific AAV promoters now. We have a capsid, and so I'll summarize this here. They're novel capsids from Mark Kay's lab. There's a series of human beta cell specific promoters, and you don't want just super strong promoters, you want weak and everything in between, and those now exist. So the next step is then to turn this beta cell story around and do this reprogram alpha cells, and we think that the way to move the preclinical data look good enough that this needs to be taken to the non-human primate next. So the big question with injection into the duct with non-human primates is it dangerous to do that if you inject a largish volume of vector? A retrograde, do you cause pancreatitis? Do you cause problems? Can you transduce? And we also need to make a human alpha cell promoter, which is in the works. So we've done one monkey with AAV9 expressing a reporter. This was done by Nakai. And basically there were some pilot experiments done to see what kind of volume a rhesus macaque would tolerate with retrograde injection. We have that figured out. And panel A, you can see a contrast agent was injected. And we get to every location in the pancreas. We also exposed the vector to the bile juice, not the pancreatic duct fluids, and AAV9, which was used here, is resistant to that and can remain infectious. And in panels B and panel C, you see in pink cells that were transduced in this monkey after an intraduct injection. AAV9 seems to hit primarily pancreatic acinar cells. But what we know from this experiment is the monkey did not get sick. We didn't have elevation in amylase or lipase. So you can go into the duct of non-human primates without causing pancreatitis, and you can get, this is an N of 1 first monkey we ever did, 10% transduction of the organ, which is, I think, pretty respectable. So with that, I'd like to conclude. I apologize for going over. The capsid work was done in collaboration with the Nakai lab and the K lab at Stanford, and some of the transcriptional control elements came from the University of Pennsylvania and from Micah Sander at UCSD. Thank you very much.
Hi, Jerome Char with Sema Therapeutics. Very nice talk. I had a question about the mice that got cured by the duct injection. Mm-hmm. Could you comment on the mass of the cells that have been transduced in relation to the normal islet content of pancreas?
So there's the duct experiments we did. And there is the duct injection experiments done by the Giddis group, where they went after the alpha cells. In both settings, the C-peptide content of the treated pancreas was completely wild type. So the islet mass had been completely restored. So it wasn't like 10% the number of beta cells as a normal mouse has. It was 100%. So you know, but that being said, I'm sure you know that only 1% of the cells in the pancreas total are beta cells.
Yeah. Yeah.
So it's about 1% of the cells total.
Okay. Thank you.
So don't you expect an immune response against your new reprogrammed beta cells in type 2 diabetes patients, the same way they kill the beta cells?
Yeah, that's why I said in the beginning, I mean, replacing the beta cells is clearly part of the therapeutic paradigm, but it is expected that if you make a really good new beta cell, that the autoimmunity will chew it up again. So this will require an autoimmune suppression component as well. But it's like you can't work on both things simultaneously. I mean, they're totally different. But one of the things that people are thinking about in terms of gene therapy, we heard a little bit about this yesterday from George Church. You may be able to make reprogrammed cells immune stealthy by including things in the AAV that makes it so that those cells are actually less immunogenic.
And do you see any effect on some of your quiescent islets that exist in the pancreas? Not necessarily those that are active near the ducts when you actually do this intraductal?
Like proliferation or?
No, just you start turning on the insulin genes in these cells, or creating or turning on the genes within these quiescent islets?
Yeah, so we basically, I didn't want to go through all the controls. In our duct reprogramming, we really didn't, it seemed like the adenovirus, probably because of its size and so forth, was really limited to transducing only the ducts. I think that would be more of an issue with AAV.
With that, we're going to close the session. Thank you very much.